Welcome to this explainer. We're diving straight into something absolutely fascinating today, the hidden machinery that makes us exactly who we are. If you've ever wondered about the invisible biological forces that are literally controlling human evolution behind the scenes, well, you're in the right place. We aren't just looking at abstract history here. We're talking about the living, breathing code inside your cells at this exact moment. Let's get right into it.
Think about this for a second. Why do some physical traits just vanish without a trace, while others completely conquer the globe? Every single quirk, every genetic advantage, or even susceptibility you have right now, it's all the result of a microscopic, invisible battlefield that's been raging for thousands of generations. Our genetics are essentially this ancient living record of survival. And today, we're going to learn exactly how to read that record.
Okay, here's our roadmap for today. First, we'll look at the origins of genetic variation. Then, the three forces of evolution, followed by reading the genomic record. After that, we'll use those rules for calculating disease risk, and we'll wrap it all up with an epic case study on lactose tolerance.
All right, moving into Section 1, Origins of Genetic Variation. Basically, how new traits enter the gene pool. Before a trait can even be tested by evolution, it actually has to exist in the first place, right? So genetic variation gets introduced in two main ways. Down at ground zero, you have molecular level variation. These are spontaneous changes. Think of them as random DNA copying errors or typos that accidentally create a brand new biological function. Then, on a broader scale, we have population level variation, also known as gene flow. This happens when individuals carrying pre-existing variants pack up and migrate into a totally new population. So it's either a brand new spelling mistake in the DNA or new code literally walking right through the front door.
Section 2. The three forces of evolution. Let's look at the fate of a genetic variant.
To really wrap our heads around natural selection, let's imagine a brand new mutation pops up and it gives someone bright blue hair. Okay, under negative selection, if having bright blue hair makes you super visible to hungry mountain lions, well, you're probably going to get eaten before you can have kids. The variant's frequency drops. But conversely, under positive selection, what if that blue hair makes you incredibly attractive to potential mates? Suddenly, fertility skyrockets, way more blue-haired babies are born, and the variant's frequency shoots up. Ultimately, it's the environment that dictates the selective pressure.
But here's a really critical nuance you have to remember. Timing matters immensely. At the end of the day, selective pressure is entirely graded on reproduction. If a genetic variant causes a fatal childhood condition like Tay-Sachs disease, that individual tragically won't survive to reproductive age. Because of that, the variant faces heavy negative selection. However, look at late-onset conditions like Alzheimer's or certain breast cancer variants. These usually show up way after someone has already had children. Because the genes have already been passed on to the next generation, they completely evade negative evolutionary selection. Evolution literally only grades you on what happens before you reproduce.
And then we have the absolute wild card of the whole system, genetic drift. Forget about mountain lions or survival of the fittest for a minute. Genetic drift is driven purely by random blind chance. Imagine a random infection sweeps through a village, and by sheer terrible luck, it wipes out most of the blue-haired people. That infection had absolutely nothing to do with hair color. It was totally independent of how biologically useful the blue hair variant was. But here is the kicker. Genetic drift has a drastically larger effect in small populations. If your tribe only has 10 people, and 2 die from a random lightning strike, you've just completely reshaped the genetic makeup of the survivors. It can lead to alleles being completely lost forever or permanently fixed in the population.
Section 3. Reading the genomic record. This is where we start decoding our history.
It all starts with something called haplotypes. Now, a haplotype is basically just a sequence of DNA, a cluster of genetic variations that tend to be inherited together as a package deal. If we want to figure out how genetically diverse or closely related a specific population is, we don't have to guess. We can estimate that diversity incredibly accurately just by counting up the number of unique haplotypes present in that group. More unique haplotypes simply means greater genetic diversity.
Now this brings us to a term that sounds kind of intimidating, but I promise it's actually really cool. Linkage disequilibrium. By definition, this is the non-random association of 2 alleles or variants in a population. But honestly, just think of it as genetic hitchhiking. When a brand new mutation pops up on a chromosome, it sits right next to other, already existing genetic markers. If that new mutation gets passed down, those neighboring markers often just hitch a ride and get passed down right along with it, staying firmly linked together over time.
This raises a really fascinating question for geneticists.
As generations go by, DNA normally gets shuffled around and broken apart through a normal process called recombination. Knowing that, if we look at modern DNA today and find an incredibly long, unbroken genetic sequence, a long haplotype block, what does that actually imply about that variant's history? Well, this perfectly illustrates one of the coolest concepts in genetics. Finding a long region in linkage disequilibrium means that variant popped up recently and then went through incredibly strong, rapid positive selection. It spread through the population so fast, we're talking a very small number of generations, that the normal shuffling of DNA literally didn't have enough time to break the block apart. It's kind of like finding a pristine, completely unbroken ancient artifact. It tells us that something massive and sudden happened there.
Let's look at Section 4, Calculating Disease Risk. We'll use cystic fibrosis and the Hardy-Weinberg model.
Cystic fibrosis is an autosomal recessive condition. That means you need 2 copies of the disease variant to actually be affected by it. It's most common in people of Northern European descent, affecting roughly 1 in 3,200 newborns. Now, by using something called the Hardy-Weinberg equilibrium equation, which models population genetics, we can take that 1 in 3,200 number and work backward. When we do the math, we find out that the frequency of the actual disease-causing alleles in the broader population is exactly 1.8%.
Okay, so once we know the allele frequency is 1.8%, we can calculate the rest of the variables to figure out the carrier frequency. And it turns out that roughly 3.5% of individuals of Northern European descent are carriers of the cystic fibrosis variant, meaning they don't actually have the disease themselves, but they are absolutely carrying that hidden genetic code for it.
Let's ground this in a real family planning scenario. Imagine a female who actually has cystic fibrosis. Because she has the disease, we know for a fact she will pass down one disease allele to her child. Now, say she wants to have a child with a Northern European male, but he has no idea what his carrier status is. What are the odds their child will be affected? Well, Step 1. There's a 3.5% chance the male is a carrier. Step 2. If he is a carrier, there's a 50% chance he passes that specific allele down. Step 3. We just multiply those odds together. 3.5% times 50% equals a 1.8% total risk of having a child with cystic fibrosis.
But let's take this one step further. What if her partner was Asian American instead? In that population, the carrier rate is drastically lower. It's about 1 in 120. If we run that exact same math, the risk drops all the way down to just 0.4%. So the crucial takeaway here is this. 1.8% divided by 0.4% is 4.5. She has a 4.5-fold relative risk increase of having an affected child with a Northern European partner compared to an Asian American partner. It's a perfect example of how ancient population history and genetic drift directly dictate our modern clinical odds.
Finally, Section 5, Case Study, Lactose Tolerance. This is a true evolutionary epic.
You know, it might actually surprise you to learn that ancestral humans were completely lactose intolerant. Back in the day, once we were weaned off our mother's milk, our bodies just completely shut down lactase production. So lactose tolerance is actually a pretty new genetic trait. And even crazier, it's a convergent adaptation. That means multiple, totally independent variants popped up in completely different populations all around the globe. And these mutations didn't even change the lactase protein itself. Instead, they happened in the DNA's enhancer region, which essentially just jammed the LCT gene in the on position, keeping it transcribing right into adulthood.
We can actually trace the earliest emergence of these lactose tolerance variants back roughly 20,000 years ago. But the latest emergences, those happened just 2,000 years ago in completely separate independent communities. And remember what we learned about linkage disequilibrium just a few minutes ago? Well, in present-day populations, the genetic variants for lactose tolerance are found in noticeably long haplotype blocks. That is the smoking gun confirming they spread rapidly and aggressively through the human gene pool.
So why did this specific variant catch on like wildfire? The essential catalyst was livestock. Listen, just having the gene mutation isn't enough. The environment actually has to reward it. For ancient communities relying on cows, goats, and sheep, suddenly being able to digest dairy provided a massive, uninterrupted nutritional advantage, especially when times got tough during famines or long, harsh winters. It sparked absolutely intense positive selection. The individuals who could drink milk survived, thrived, and passed those genes on to more children.
So, we've seen how these random mutations arise, how selection guides them, and how population dynamics permanently cement them into our biology. But remember, evolution didn't just pack up and stop a few thousand years ago. It is an ongoing, non-stop process. So I want to leave you with this final thought. If our DNA is a living, continuously updating record of our history, what hidden traits do you think are undergoing positive selection in humans right now? Thank you so much for exploring this incredible biological history with me today.
Stay curious, and I'll catch you in the next explainer.